Okay, I'm going to finish chapter 6, which I think the only thing left was enzymes. Okay. The catalyst is a chemical agent that speeds up a chemical reaction without being consumed by the reaction. An enzyme, which is a protein, is a catalytic protein. Hydrolysis of sucrose by the enzyme sucrase is an example of an enzyme-catalyzed reaction. Remember that your enzymes are going to end in ASE. So here's sucrose being broken down by water, so this is a hydrolysis reaction. Sucrase is the enzyme that catalyzes that reaction, breaking down sucrose into glucose and fructose. Every chemical reaction between molecules involves bond breaking and bond forming. The initial energy needed to start a chemical reaction is called the free energy of activation, or activation energy. Activation energy is often supplied in the form of thermal energy that the reactant molecules absorb from their surroundings. So here is an energy diagram where we have our reactants here. Our transition state is when the bonds are breaking. This is the activation energy, the energy required to start this chemical reaction. And then these reactants get converted to products. Now in this case, this is an exothermic reaction because the free energy of your products is less than the free energy of your reactants, or delta G is less than 0 or negative. Enzymes catalyze reactions by lowering the activation energy barrier. Enzymes do not affect the change in free energy delta G. Instead, they hasten reactions that would occur eventually. So they speed up chemical reactions by lowering this activation energy right here. Well, this would be the activation energy without an enzyme. And then if you add an enzyme to the reaction, it lowers the hill that the reaction needs to get over in order to proceed. The reactant that an enzyme acts on is called the enzyme substrate. The enzyme will bind to its substrate, forming an enzyme-substrate complex. And then the active site is the region on the enzyme where the substrate binds. And remember that enzymes are very specific to their substrate. Enzymes change shape due to chemical interactions with the substrate. The induced fit of the enzyme to the substrate brings chemical groups of the active site into positions that enhance their ability to catalyze the reaction. So this would be an enzyme and the active site, which is a groove within the enzyme where the substrate will bind. And then when the substrate comes and binds to the active site, the enzyme sort of surrounds the substrate even more, sort of if you were shaking someone's hand, how your two hands sort of mesh together or come together in a tight fit. In an enzymatic reaction, the substrate binds to the active site of the enzyme. The active site can lower an activation energy barrier by orienting substrates correctly, straining substrate bonds, allowing them to start to break, and providing a favorable microenvironment and covalently bonding to the substrate to hold it in place until it can be converted to product. Okay, so here we have our substrates, our reactants entering the active site. That forms the enzyme-substrate complex, and then those substrates or reactants get converted to product. The products get released, and then the enzyme gets reused. An enzyme's activity can be affected by general environmental factors such as temperature and pH, chemicals that specifically influence the enzyme. Each enzyme has an optimal range of temperature and pH in which it can function. Optimal conditions favor the most active shape for the enzyme molecule. So basically what that means is the active site will be the correct shape in order for the substrate to be able to bind to it. Okay, so on the top, we have two reactions. We have two different enzymes. One enzyme is typically found in humans. It's a typical human enzyme that functions its optimal temperature is 37 degrees Celsius, which is our internal body temperature. Then if we look at this second enzyme, this is an enzyme found in a thermophilic bacteria which has a temperature of 77 degrees Celsius. So you can see that the two enzymes have very different optimal temperatures. Okay, so as we go outside the range of the optimal temperature, the rate of the reaction starts to rapidly decrease and then goes to 0. And then here is optimal pH for two different enzymes. This is pepsin, which is a stomach enzyme. The pH of your stomach is about 2, so the optimal pH for this enzyme is 2. And then this is the optimal pH for trypsin, which is an intestinal enzyme. So the pH of your intestines is about 8. So you can see very different optimal pHs for different enzymes found in different places. Cofactors are non-protein enzyme helpers. They may be inorganic, such as a metal in ionic form, or organic. An organic cofactor is called a coenzyme, and coenzymes include vitamins. There are also what are called enzyme inhibitors. Competitive inhibitors bind to the active site of an enzyme and therefore block the substrate from binding there. Non-competitive inhibitors bind to another part of the enzyme, which causes the enzyme to change shape, and it changes the shape of the active site as a result. Examples of inhibitors include toxins, poisons, pesticides, and antibiotics. So here's a substrate which will normally bind to the active site of the enzyme. This is competitive inhibition, where you have some other molecule binding here, blocking the substrate from being able to bind there. This is a non-competitive inhibitor. It binds somewhere else on the enzyme, causing the shape of the active site to change. Therefore, the substrate can no longer bind there. Enzymes are proteins encoded by genes. Changes or mutations in genes lead to changes in amino acid composition of an enzyme. Altered amino acids in enzymes may alter their substrate specificity. So in other words, the proteins, the enzymes will not be able to bind to the substrate that they're supposed to bind to because their active site shape is different. Under new environmental conditions, a new form of an enzyme might be favored. Chemical chaos would result if a cell's metabolic pathways were not tightly regulated. A cell does this by switching on or off the genes that encode specific enzymes or by regulating the activity of enzymes. Allosteric regulation may either inhibit or stimulate an enzyme's activity. Allosteric regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. Most allosterically regulated enzymes are made from polypeptide subunits. Each enzyme has both active and inactive forms. The binding of an activator stabilizes the active form of the enzyme, and the binding of an inhibitor stabilizes the inactive form. Okay, so here we have allosteric activators and inhibitors. So here we have an activator that's binding to the enzyme. This reinforces the shape of the active site so that substrate can bind to the active site. Here we have an inhibitor, which is bound to somewhere on the enzyme, which causes the shape of the active site to change. So this stabilizes the inactive form of the enzyme because the substrate can't bind here. Cooperativity is a form of allosteric regulation that can amplify enzyme activity. One substrate molecule primes an enzyme to act on additional substrate molecules more readily, so it increases the effect of the enzyme, the activity of the enzyme. Cooperativity is allosteric because binding by a substrate to one active site affects catalysis in a different active site. In feedback inhibition, the end product of a metabolic pathway shuts down the pathway. Feedback inhibition prevents a cell from wasting resources by synthesizing more product than is needed. So this would be an example of feedback inhibition. So here we have the production of isoleucine. And isoleucine, what it does is it binds to the first enzyme in the pathway, and it shuts off that enzyme, thereby shutting off the whole pathway. So again, this is done to save resources and energy. Structures within the cell help bring order to metabolic pathways. Some enzymes act as structural components of membranes. In eukaryotic cells, some enzymes reside in specific organelles. For example, enzymes for cellular respiration are located in the mitochondria. So this is important because you want to have the enzymes for cell respiration in the mitochondria. You don't want to have them somewhere else, like in the cytoplasm, because that will reduce the efficiency of the reaction.